This is a production of Cornell University.
So thank you so much to Olena and Mike and the organizers. It's a real joy to be here. It's my first trip to the Mecca of plant science in the US, so I'm really, really excited. So before I show you some of the phenotyping data, let me explain what the Plant Imaging Consortium is. So we are a consortium of Arkansas and Missouri-based scientists. We're funded by the NSF EPSCoR Track 2 program. And basically, we are a group of plant scientists who came together convinced of the importance of plant high-throughput phenotyping and other imaging tools. And what the project allowed us to do is really open the access to some interesting high-throughput phenotyping and other imaging technologies to really empower discoveries in plant stress biology. So I want to acknowledge some of my colleagues. There are 3 scientific leads in Missouri and 2 in Arkansas. In Missouri, the leads are Sylvia Jurison, Joan Chuantai, and David Braun. And in Arkansas, the leads are Fiona Goggin and myself. And again, I'll tell you more about it. This is our website, and among the things that we have done is funded young scientists, and I learned yesterday that you are getting an excellent one coming back to Cornell, so that's wonderful. You are going to see also here Noah Folgrin and Malia Gihan. These are 2 very talented scientists, and they got some funding from our consortium to really develop further PlantCV, which is an open source software, and it's really a community resource where you can contribute, and I'm going to show you what we have contribute to this field. Okay, so I'm pretty sure that I'm preaching here to the choir. All of these abiotic stresses and also, of course, biotic stresses present a big challenge to agriculture, and we need better ways to measure plant performance and this interaction, the genes by environment interaction. To do that, we are convinced, and I believe Todd's talk already showed us, digital phenotyping is a very powerful tool because you go beyond the resolution of the human eye to really extract all of this important information out of these images. So let me discuss the platform. So in Arkansas, there is 50 plant scientists throughout the state. You have here a treasure, you have 80, right? We are 50 and we are scattered in 5 different universities. So we came together and said, how can we be more impactful? So we came together and formed a virtual center, right? So we call it the Plant Power Production. And again, with NSF funding, we were able to acquire this instrument because we were really looking for things that will empower us and do go from descriptive science to quantitative science, and go from a very, very biased approach, which is manual phenotyping, to an unbiased approach. So this is the system we acquired. It's a Scanalizer HTS from Lemna Tech. It has 4 cameras, a visible camera, a fluorescence camera, an infrared camera, and an infrared camera, and already told us some of the benefits of each of these sensors. And what I want to bring to your attention is we can phenotype Arabidopsis, we can phenotype also the seedling stage of many crops in this configuration, and we also have a deck that accommodates plates and multi-well plates. So the downside of our system is there is a finite space here, so I cannot go beyond the before the vegetative state of many crops. So that's the downside of the system we have. Okay, so again, a little bit more detail on the cameras. The visible camera, again, the workhorse. You can interrogate and get information regarding size, architecture, and color. With the fluorescence camera, we can have an indication of plant health through chlorophyll fluorescence. Or with the special filters, we can measure fluorescent proteins or for example, in hairy roots that are non-photosynthetic, we can measure phenolics. With the re-infrared camera, we can get information regarding water content, and with the infrared leaf temperature. Okay. So again, why do we like it? It's unbiased, non-destructive. Several phenotypes can be detected, and this is one of my favorites. Raw images are permanently stored, and you can come up later with a different question and interrogate that data set. Now, if careful metadata is kept, I think there is a lot that can be gained by the overall community if we deposit this data in places where it's public, where Olena can interrogate my data, I can interrogate Chris, and so on and so forth. So the throughput of our system is, if we do multi-well plates in the order of 8,000 samples per run, and for most of the assays we do with plants in soil, between 18 and 45 plants per run, and a run usually takes less than an hour. So I love the system, mostly because experiments that before it took me 6 months to get done, now I get them done in 4 weeks. So, and also, now I can manage a lot higher number of samples, and then the statistics and the robustness of your data improves. Okay, so this is my current team. Half of the team does vitamin C metabolism, which is my passion. I love plants and biochemistry of plants, but the other half of my team really does phenotyping for several universities in Missouri and for all of the external users we're serving. This is a very talented postdoc, Dr. Sujin Liu. He's the one who has developed all of the algorithms applications that I'm going to show you. And again, very talented group of students. So the plants we have the most experience with are shown here. And I'm going to, Olena asked me to show an example with Arabidopsis and one with a real crop. So I'm going to show rice today. So here are my examples. So to show you what we have done, or one of the projects we have done in Arabidopsis, I'm going to show you a collaborative project with the laboratory of Dr. David Mendoza from the University of Missouri, Columbia. He's an ionomics person. He's into loving metals, like several of you here. So he loves metals and understanding how metals are absorbed and then transported. So what we have done in this article, in this paper that we published recently, is really do ionomics and phenomics in the same plants under water limitation conditions. So basically, here is the experiment. We germinate Arabidopsis seeds in plates. We select the robust seedlings. We transfer those to soil. Once we have robust seedlings, we divided them into 4 different treatment groups, and a control group and 3 ones with water limitation conditions. And the plants were phenotyped every other day. Of course, we measure, we make sure that we are imposing water stress. And in addition to the phenotyping with the scanalizer, we are also incorporating this beautiful instrument developed by David Kramer's laboratory at Michigan State. It's called Multispec Q. It's a non-destructive instrument that allows you to do the same thing that a Lycor does, but instead of costing you $60,000, it costs $1,000. So again, what we like is non-destructive, and we see great correlation between what the data we're capturing with this instrument and the fluorescence data we're getting with the scanalizer. And of course, the ionomics person loves his ICP-MS, right? As you can imagine. Okay, so here a little bit of the images. Again, control and 3 water stress conditions. We're showing you the images and the analyzed images. All of this data set was analyzed with the LemonGrid software that we acquired with the instrument. And as you can see, really the big changes in size of the plant come until you get to these levels of water limitation. So here is the big surprise for us. So Arabidopsis is very resilient, and you can get down up to 50% water, and there is no penalty in the growth of the vegetative growth of the plant. However, look at what happened to seed yield. Even at this level, big drop in seed yield. So what we found out is seed yield is a lot more sensitive to water stress than vegetative growth. So in terms of the ionomics, what David found is that the effect of drought is very element and very treatment specific. And again, I know several of you metal lovers will bombard me with questions, so I just wanted to illustrate some of the power of this. Okay, so something that Lucia Costa, the PhD student that did this work, noticed is that although from the images from the top there was no difference in leaf area between these two treatments, she noticed that there were more leaves here. And that's where Dr. Liu developed for us this beautiful strategy to count leaves more effectively. And indeed, we found that under control conditions there were more leaves. This module for leaf counting has been added to Plant CV. And again, that's, I really encourage you to take advantage of this great resource. You can apply it to images that you acquire within a system. You don't have to necessarily take it with any particular system. It's really flexible, really good. Okay. So again, I couldn't not talk about Starving Biosynthesis. So this is a molecule that is my big passion. I've worked on it for the last 14 years. It turns out that plants have 4 different ways to get to that molecule. And I was very fortunate to be part of the team that at Virginia Tech discovered this pathway that starts with myoinositol. So since I moved to Arkansas State, my students and I now have characterized at least one member of each one of these gene families. And when we overexpress them, and here I'm showing you what happens when we overexpress the first enzyme, this happens to plants. These plants are the same chronological age, grown under the exact same conditions. This is the high vitamin C one, and this is the wild type. So this looking for a way to quantify this phenotype is what got me into phenotyping. I wanted to go from pretty pictures to, okay, tell me how much bigger this is. Okay, so at the beginning, like all of you, we did lots of manual phenotyping. And what we have done is now validated that all of the manual phenotyping we did was indeed correct, but now we have new readouts of health that we didn't have before. So here I just want to clarify, this is the wild type, this is the high vitamin C line, and I'm using as controls two lines that overexpress a proton pyrophosphatase. This line is very famous and used as controls because it's a known salt-tolerant and drought-tolerant line. So as I told you, color classification is very powerful, but we were sort of limited with the lemna grid approach because we were only able to distinguish green and yellow, but not different tones of green, which is what we wanted to do. So again, Dr. Liu developed a color classification approach that has given us much better resolution in understanding. So this is what happens to wild type in response to salt, and the transgenics, the highest score rates don't show this yellow in phenotype. That is, I should clarify, is not noticeable with your naked eye. Okay, so I'm going to switch now to rice to show you an example with rice. So this is funded by the Plant Genome Program, and it's a collaboration with the University of Nebraska-Lincoln. So we have looked together for novel sources of salt tolerance in rice. And my main collaborator is Harkam Alwali, and he has done all of the phenotyping at the tillering and the panicle initiation stages, and I've done the one at the seedling stage. So let me show you, and this is, I don't know if Susan is in the audience. This is very familiar to you because it's the rice diversity panel that Susan McCush and her collaborators developed. This has been a wonderful resource for us to find those novel genes involved in salt tolerance in rice, an important an important problem in the world, and certainly in Arkansas, the main producer of rice in the US. So the beauty of this is it has a core group that represents all 5 different subgroups of rice, and of course, there is all of this genomic information that Susan and her group have made available. So phenotyping this was an obvious choice. We're going after tissue tolerance, not after ion exclusion as mode of action for salt tolerance. And the very beautiful work that Harkamal has done is he did all of this work at the Australian accelerator, again, and develop a pipeline for the visible images and a pipeline for the fluorescent images. I should emphasize for the fluorescent images, a very talented undergrad in his lab wrote this software that is also very powerful for analysis. It's called Image Harvest, which will be, I guess, the cousin or of PlantCV. So this is another approach of an open source software to analyze this type of images. Harkamal already has found candidate genes and is already validating them. And again, this was at the tailoring stage, and here's his publication. Let me show you a little bit what I have done at Arkansas State. In my case, with minority undergrads, 14 of them, knowing nothing about science, they have come to my lab for the summer and they have generated publication quality data. I'm very proud of them. Okay, so Harkamal sent the seeds, we clean them, we germinate them. Once we have healthy seedlings, we transfer them to this hydroponic system. This is just calcinated clay to provide support. We put these phenotyping trays in these tubs, and this is where we put Yoshida solution as a control group, and and we set up an identical tray, but with salt. So in the first experiments we did, this was our control group, and we were providing the salt in 3 different steps, but we were not seeing a robust phenotype, so we went a little bit more aggressive. So in all of the data I'm going to show you, this is the black line is our control, and we made the salt in 3 increments and phenotype during the experiment. So first let me show you the result with a known salt-tolerant line. This is the representative images of the line treated just with Yoshida solution, and this is what happens. This is really a salt-loving line. Our digital readouts completely back what the images say, and manual phenotyping also backs what we saw with the salt-tolerant. This is an example of a salt-tolerant line. Again, this line grew better than the control, and the manual phenotyping we did at the end of the experiment fully backs up these results. And as you can imagine, most of the lines were salt sensitive, where the treatment grew a lot less than the control. And again, the manual phenotyping fully backs that. And the interesting thing is that although your eyes cannot see it, the instrument was able to tell us there was a lot more yellowing in the plants treated with, in this case, in the salt-treated ones versus the control. Okay, so although I'm showing here just a group of the lines, not all of them. Again, these 14 students have allowed me to now phenotype 200 of the lines of the rice diversity panel. We have gone through the analysis of 84,000 images. As Todd was saying, very big data sets. You need to develop a different set of skills to handle that amount of data. And I just want to finish telling you that we are a core facility. We accept new users. And whether you already have the money to do the experiment or are thinking about getting the money to do the experiment, we are happy to talk to you and to help you. We are a resource for the plant science community. And we hope you find this useful. My colleague, Dr. Joan Tran Tai, wants me to tell you that he has this beautiful PET imaging system at Washington University at St. Louis. He's an incredible scientist. He made this on his own. It's really, really amazing. And Margaret already showed you the power of this technique, and I'm sure Chris will share some of the power of this. To me, as a biochemist, it's just so amazing to see how the carbon that was given in the leaves suddenly can be monitored in a plant that is still alive, right? It's just like mind-blowing. Okay, so my closing slide is this one. I just want to tell you I really believe there is the need for more cooperation. I'm very grateful to the opportunity to be here. I hope there is a lot of opportunity for interaction, collaboration, etc. And I encourage you to get very much involved in the North American Plant Phenotyping Network. This is a group of people that we are all enthusiasts about phenotyping and about learning together and making of this a more vibrant community in the United States. So please contact me or any of my colleagues involved on this. And with that, I'll be very happy to take any questions.
Questions? Good talk.
I'm just so surprised with the, you showed us images that the Lemnatec, Throughput didn't allow you to distinguish just green and just two colors, and then your own in-house. I don't know, what did you do so that you was able, what did the software, or like what did you change there?
Well, we really want, so this is our dream, okay? Our dream is, or not only dream, as one of the goals of the Plant Imaging Consortium is to develop a database where we can all put the data, but we want to have also the tool to allow people to analyze their data. So we really wanted to write things that were compatible with PlantCV, okay? Because we really think an open source software will be a much easier way to handle things. So my postdoc wrote completely a new algorithm. Yes.
So the sensors were the same?
The sensors were the same, the images were the exact same. Yes.
One more question before we move on? You can always add this at the break, which will begin at 10:30 after my talk. All right, let's.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.